Welcome to day two of our 29th annual IWMF Educational Forum, and we're just so excited to have over 850 people from throughout the world joining us in person here in this room and virtually. I'm Newton Guerin, and IWMF President and CEO, and on behalf of really all of us at the IWMF, thanks to each of you for being with us. This morning, I want to acknowledge our 2024 Ed Forum sponsors for their financial support that really helped to make this event possible. First, we'll start with our supporting sponsors, the Leukemia and Lymphoma Society and Dana-Farber Cancer Institute. And both of these incredible organizations have been, in addition to providing financial support for us for this week, have just been incredible partners for the IWMF in so much of what we do throughout the course of the year. So let's give them a special round of applause. Our premier sponsors, Selectar Biosciences and Pharmacyclics and Johnson & Johnson, we appreciate their continued support. I know that there are a number of their folks, several of their folks that are with us today and here in the ballroom, and I'd like to ask them to stand so we can say thank you in person. Selectar and Pharmacyclics, Johnson & Johnson, Michelle, thank you so much. And finally, I would like to recognize Beijing for their sponsorship gift of $100,000. And by the way, this is their second year to support the Ed Forum at this giving level. So we are so appreciative of their support. And it's my pleasure to introduce Dr. Todd Zimmerman, who is Beijing's scientific medical director, who is going to be with us to just tell us a little bit about what's going on in Beijing. So, Todd? Well, you come up, I'll come down.
A little dance there to get the morning started. So not as good as the dance last night with the West Coast swing. So you'll not see me do that. So. But it really is a pleasure to be here this morning. So by way of introduction, I'm Todd Zimmerman, a hematologist by training and senior medical director at Beijing on the U.S. side. Prior to being at Beijing, here I had a prior career where I was a practicing hematologist oncologist at the University of Chicago. And when I started my training there in 1992, Waldenstrom's was a thing, that we had heard about, but it was, poorly understood. It was frequently misdiagnosed, and then when you had a diagnosis, there weren't really a lot of treatment options. But fast forward now to when I got home earlier this week here, I opened up my copy of Blood, which is a journal here, a research journal too, and one of the premier research articles was a review of how we can use molecular genetics here to define which treatment someone with Waldenstrom's should get. So just in that time period, it's been amazing what's happened in there. And that didn't happen by chance. I look around this room here, I can see brilliant scientists, excellent clinicians. I see a number of pharma partners that have made this all possible. But without the glue and that thread of the IWMF pulling everything together, it probably wouldn't have happened. Because everyone seems to work in silos here, but you have great organizations like the IWMF that pull everyone together here and say, all right, let's focus, let's all pull in the same direction here. And that's how we're getting to where we are. And this is the big reason here. We're here to support the IWMF and the programs that they do, like the Educational Forum and the workshop that will be conducted this fall in Prague to really kind of advance research. Because really, as far as we've made it here, we still need more research. And that research really comes through, a number of different collaborations. We've been proud of the work that we've done at Beijing with the development of Brukinsa or zanubrutinib through the Aspen study, and it's been very heartening to hear a lot of your stories about how you've been on it here and how you're tolerating it here. But we know that there's more that we can do. We have active research programs here where we're looking at combining zanubrutinib with other drugs. Can we make the course of treatment shorter? Can we make the treatment shortened better here by adding other agents to zanubrutinib here for patients with Waldenstrom's? And then we also have new drugs, too, that we're looking at, and we're specifically going into the Waldenstrom arena to take a look at that. One is our BCL2 inhibitors and Rotoclax. So BCL2, so essentially cells should have a kill switch. So interesting fact here, the reason we don't have webbed fingers here is because the webbing between those, the skin in there had a kill switch called apoptosis that kills that during the development process. But also, when you think about a cell that has too many genetic abnormalities in it here, it should die. And so there's this automatic kill switch called apoptosis, and a lot of times cancer cells don't do that. So we have a drug that we're developing, the second in class, that will hopefully improve that and kind of, be able to work on killing those cells here that should be dying. And that's called sonrotoclax, and there's an active development program in Waldenstrom's for that. The other compound that we're very excited about is our protein degrader, our CDEC as we call it. A protein degrader, essentially, I like to think of it this way. If the door opens up here and lets things happen here, and there's the key, the BTK inhibitors like zanubrutinib go in here and they block it so you can't open that door. Eventually what happens is a lot of times these cells learn to kind of get around that, and then you can still open the door here because they become resistant. What a protein degrader does here is it gets rid of the doorknob. So you're not going to open that door here because we just completely destroyed that protein. And so that's an active program that we have going on in a different B-cell malignancies, including Waldenstrom's macroglobulinemia. So we're very excited about the opportunities that we have there from that standpoint. But we're also thinking about what's really at stake here, and that's you, the patients and the caregivers. I think someone asked some of us this last evening here why we're here, and we're here to meet you because you're our inspiration. You're the reason that we do what we do so that we can make life better for everyone. And at Beijing, we're really committed, and our patient is really kind of our North Star when we look at how we want to face things. So even when it comes to designing a clinical trial, we involve patients and their caregivers early on when you develop the study here. If we're developing a study here where the endpoint isn't meaningful to you here, we shouldn't go down that route. If we develop a study where it's too cumbersome here and it's going to really impact your quality of life too much, we shouldn't go down that route too. So early on in the process here, we want to engage you here and make sure you're involved in that process. There's other ways too that we support patients as well. We're a presenting sponsor for LLS Light the Night. That's a phenomenal thing. If you've ever been to it here, if you haven't been to it, I would look it up here for this fall to see this sea of balloons with people out there celebrating survivorship. Remember those who didn't make it too. It's really, truly a moving experience here, an important part of the LLS mission and what we need to look for. So I've been talking a lot about hope and the hope that we have. But the truth is that when you're faced with a diagnosis like Waldenstrom's or something serious, you're faced with a lot, and that can affect your mental health. It's completely normal, whether you're the patient or the caregiver, to be hit with depression, to be hit with anger. And we recognize how important it is for the mental health of patients who are facing these severe diagnoses. So as a company, we've started out a Talk About It campaign where we're focusing about the mental health and the importance of mental health for patients with malignancies. Doesn't matter what malignancy you have, it's open to everyone here, and it's for both patients and caregivers, because it's important that everyone be able to talk about it. Because while there is hope, there's still the reality that we're facing something serious too. So again, just in closing, I want to thank all of you for being here today, because you really inspire all of us to kind of want to go to the next step here and make things better. I have a small little kind of statue that sits in my desk of a globe and a person sitting on it with kind of his heads in his hand, and it's called Until There Is a Cure. And that's why we're here. Until there's a cure here, we're not going to be done. So thank you very much.
Thank you, Todd and Beijing, and thanks again to all of our sponsors. We all appreciate everything you do. So one last round of applause. There we go. It is now my special pleasure to introduce Dr. Steve Ansell. Steve is one of those people where words really can't adequately describe the impact that he's had on the lives of the global WM community, that he has had and that he continues to have. Dr. Ansell has really dedicated his career to improving patient outcomes while leading a global effort to get to a cure. He currently serves on the IWMF Board of Trustees. He co-chairs our Scientific Advisory Committee along with Dr. Steve Treon, and he serves as the chair of our Research Grants Review Committee. And for those of you who are not familiar, that's the group of WM experts from throughout the world who reviews on an annual basis many grant applications. I believe we have 22 in this most recent cycle. They'll review those grant applications and then make the determination about which grants should be funded. So it's an incredible undertaking, extremely important, and they do a fabulous job. If you haven't had a chance to read Dr. Ansell's very impressive bio in our program guide, in your program guide, please do that. And with that, I will turn things over to you, Steve. Thank you.
Well, good morning, everybody. I'm not entirely sure what to do with words can't express what he does, because maybe the words are, not to be repeated in mixed company. I'm not exactly sure where that one was going, but thank you, though. So my job this morning is really to keep everybody here abreast and updated on where things are going with the IWMF-funded research. And I really appreciated what Dr. Zimmerman just spoke about when he was talking about where things were and now where things are. And I'm going to just highlight a few of those things and give you some sense of what the research is really all about. A little bit later on, you're going to meet two of our junior faculty who are coming into this field, and they're going to share a little bit about themselves. But it's just super exciting to see how the field has moved forward, and that is directly because of the contributions of the IWMF, and particularly people like you. So just wanted to right off the bat say thank you so much for what you do. So when I started in this field 20 years ago, it was really interesting. Todd mentioned this. We actually had a meeting where we all got together and we said, what's going on in Waldenstrom's? And the answer was, this meeting can be super short because the answer is nothing. And quite frankly, most of the meeting was then talking about what we didn't have. So the first thing we didn't have was any kind of model. There was no mouse model. There were no cell lines that really in any way represented what was going on in Waldenstrom's. And why is that important? Well, you need to sort of create a premise. You need to understand what you need to research by using something that you can do a lot of testing in. You can't do a lot of testing in people. You need to do it in cell lines and in animal models. So did we have those? No. So that was a problem. So right there was a discussion about we need to develop that. Second thing was, what do we understand about Waldenstrom's? And the answer was very little. And why was that? Well, because it was an uncommon disease, and everything we thought we knew, we had really just borrowed from cousin diseases. We'd either borrowed it from multiple myeloma, or we'd borrowed it from chronic lymphocytic leukemia, or we'd borrowed it from lymphomas, but everything was just extrapolated. Very little was, we know this for a fact to be true in Waldenstrom's. The third thing was, there were no researchers working in Waldenstrom's. So I think at that time, maybe Steve Treon and I were somewhat interested, but aside from that, and I think Irene Gabriel, I need to remember who was in that meeting, but not a lot of people. And basically, because not a lot of funding, not a lot of focus on the whole area of Waldenstrom's. And you heard Dr. Zimmerman mention this, when you said, well, how are we treating patients with Waldenstrom's? It was with leftovers. We basically just borrowed stuff from other diseases and gave patients with Waldenstrom's drugs that worked in myeloma or worked in lymphoma, but very little evidence that they were specifically good in Waldenstrom's. So that's really where you became part of the story. So enter the IWMF. And right back at that time, at that particular meeting, was a case of we need to start with the basics. we need to provide tools. You can't actually build things if you don't have bricks. So let's say, what are the bricks we need scientifically? And things like, we need cell lines. So there was an initiative to say, apply, and we can provide funding that will actually allow you to develop cell lines. And many of you will know there are not a lot of them because it proved to be super hard. Again, one of the things that's pretty unusual is Waldenstrom cells seem to grow really well in people and really badly in a test tube. Why is that? Well, we need to work that out. And so that's part of what the discussion was about. Could we at least find some cells that would grow in a test tube so that we could then use them to actually decide on drugs and biology, etc. That then led to the roadmap. And I'm going to talk a little bit more about what exactly the roadmap is, but many in this room are very familiar with that. But the first roadmap meeting, nearly 10 years ago, in partnership with the Leukemia Lymphoma Society, was to say, what are the key questions we need to ask? So again, we're now focusing our attention, because we have some tools in hand, on what do we need to focus on, what do we need to work on that would actually allow us to move the field forward. And that roadmap has now been repeated, and in fact, just last month, leaders in Waldenstrom's all got together and again discussed a review of the roadmap to say, where have we been, where are we now, where do we need to go, and what do we need to do? And just to highlight the fact that this has made a dramatic difference to what we know, to the point that Dr. Zimmerman made. So what does the roadmap have in it? Why would you care about it? Well, quite frankly, it has made a profound difference to our understanding of Waldenstrom's. When we used to have ed forums 20 years ago, pretty much we had a very simplistic discussion. Didn't know much, but here's some drugs, we've been trying some stuff. That was kind of the end of that. Now, as you know, it's complicated. We're talking about MYD88, we're talking about CXCR4, we're talking about various HCK pathways, we're talking about bispecific antibodies, CAR T cells, you name it. And where did all of that come from? It came from the roadmap. So interestingly, the roadmap still has 5 focus areas, if you like. The first one is signaling. Now, you heard Dr. Zimmerman talk a little bit about the key and the doorknob. Well, that's kind of the way in which the cell gets messages. And when it gets the message, those messages going into the cell are keeping it continually stimulated. And because it's being stimulated, it starts making way more protein than it needs to make. And it grows way faster than it should. I joke and tell people it's a little bit like that annoying kid in the neighborhood that thinks it's a joke and is pressing your doorbell, ding, 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 and then runs away, comes back, ding, 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 you know, that kind of thing. And then you're super annoyed, and the whole family's like, where is the kid? You know, it's sort of on ready. And you can imagine that's what your Waldenstrom cell is doing when it gets this continual irritating stimulation. So what's the best way to stop the kid bugging you? Unwire the doorknob. In other words, when he can press the button all he likes, it doesn't do anything, right? So a BTK inhibitor pretty much does that. But that work came out of understanding the pathways that are central to the cell. There's a lot more to be learned because, interestingly, cancer cells never rely on one thing only. They have multiple ways in which they want to actually hear messages. So we have a lot to learn about other pathways, toll-like receptor pathways, PI3 kinase pathways, ERK pathways, STAT signaling pathways, etc. Number 2 is the whole question of immunology and immunotherapy. So what does that mean? Well, interestingly, a cancer cell does not live in the bone marrow or in tissue all by itself. It lives in a neighborhood, and there are other people in the neighborhood. And those of you that have heard me talk about this before would tell you that all it takes is one crack house in your neighborhood, and the neighborhood goes to pot, in no time. And that's it. The neighborhood is being spoilt. And interestingly, the actual people that are supposed to control bad behavior are not doing their job. So the immune system is standing around watching the bad behaving cells, not killing them. So how do you fix that? Well, there are a few things we need to know that we're still learning. Why is the immune system ignoring the cancer? 2, if you could get it to see it, would it kill it? Does it have the ability to do that? And then how do you actually get it mad and angry so the immune system would kill the cancer? Those are all key questions that we need to know. And many of you will be hearing more and will know bispecific antibodies that grab onto the cancer and grab onto the immune system. CAR T cells are immune cells that we're taking to training camp to get them to work better. Those are all therapies that are coming now to Waldenstrom's. So that's the therapy part. But the environment part is the other part of that immune system. What is wrong with the neighborhood that it promotes the cancer growth and doesn't get rid of it? So there's another part of that. I told you that when we were trying to make cell lines, if you took the cells out of the patient's body, put it in a dish, they didn't grow very well at all. In the patient, they were growing just fine. In fact, they were growing way too well. So the question is, why is that? Well, there's all kinds of miracle grow in the bone marrow. The key question is, what is the miracle grow? Because if there's miracle grow that's making the cancer grow, let's get rid of that. So that's what the microenvironment is all about. So what does omics mean? That's the fourth one. So there are multiple ways in which you can look at the same thing and actually understand it in a far greater and more complex way. So one of the challenges is if you only look at one thing at one time, you ignore all the other things that are happening at the same time. This is like a busy city, if you like, in your bone marrow. Cars are going by, cyclists are coming, pedestrians are coming, the light's changing, there's someone yelling, there's an ambulance going by. If all you're interested in is the ambulance, while you're looking at the ambulance, the bus will run you over. So bottom line is you need to be looking at all of these things at the same time. So what can we learn about genomics, epigenomics, transcriptomics, metabolomics, proteomics, all of that, immunomics, all of that mixed in at the same time? And then lastly, we have an opportunity in this disease, and that is people get an elevated protein before they get Waldenstrom's many times. And the key question is, some people, it just sits there for years. Some people, it never turns into anything. That's why it's called a monoclonal gammopathy of uncertain significance, because in some people, it doesn't become anything significant. But in other people, it becomes something super significant, namely Waldenstrom's. So the question is, why? And why do some people have problems and other people don't? Why do some people have problems right away, other people have it for decades and then get problems? So we could really have an opportunity to intervene. If we knew you were going to run into trouble, we could stop that happening, or we should know how to stop that happening. That's part of what that's all about. So how do you do that? Got a great idea, got the tools in place, working on the principles, but as with everything, it takes support. It takes money. And so there are 5 different areas that the Waldenstrom's Foundation, you guys, are supporting research. And I wanted to highlight briefly what they are and why they're different. So the first thing is what is called the Strategic Roadmap Initiative Grant. Then there is an Enhanced Research Roadmap Initiative. There are the Kyle Career Development Awards, and you'll hear from 2 of our recipients a little later, in just a few minutes. There is a Seed Money Initiative, and there are companion initiatives. And I'm going to take a minute just to talk about what those are and why they're different. So first thing is the Strategic Roadmap Initiative. So this is something done in collaboration with the Leukemia Lymphoma Society. Comes out of that workshop that we have all the time to define those different pillars that I just spoke about. And the goal is to say not just what can we research, but which of those things would actually take a logarithmic leap toward curing this cancer. So how do we need to reposition resources to go after that? And as I said, it focuses on those 5 areas. It also allows you to partner the research with a clinical trial. So if you're going to do correlative studies, let's say you're giving an inhibitor of the doorbell, BTK inhibitor, and you need to understand how by turning off the wiring and the bell stop ringing, does the cell and the family calm down? So if we need to understand that, we need to do some studies along with giving the patients the treatment. And that's why if you're on a clinical trial, they're drawing blood and taking bone marrow, because they're really doing that work. But someone then needs to fund the correlatives that need to go in to understand that. So pretty much this supports people for 2 years, and currently we're supporting 8 projects, and as you heard, Thursday we'll actually adjudicate a whole bunch of others that have been submitted to see which ones we feel are worthy, and we really encourage people to work together. So that's kind of the big money, if you like. But then there's the, you've got a great idea, but we're not sure it's going to work kind of approach. But it would be great to test it out. And this is what we call a seed money initiative. And the idea is here, you've just got a good idea, and we're going to give you some money to test it. Should it work? Should it look promising? You could then roll it into one of those roadmap grants which would follow. This is now the very big idea. So if you said a whole bunch of institutions would work together to say, let's answer a big question, IWMF will support usually one of these approaches where basically institutions working together gets money now not just for 2 years, but up to 3 to 4 years, the idea being to really propel the research forward. And then what we're going to focus on in the next few minutes is people like me and Dr. Treon and others that have been in the field now for, I don't know, 30 years or something, we're getting to be old codgers. And it's time to bring along the next generation. So it is a massive mistake if all you did was have you do the work and nobody came along with you. So key initiatives, and you'll hear about this one and then one more, is it's all about the next generation. And Dr. Kyle was one of those people who really understood that. And you actually saw him, nearly 100 now, on the Zoom call yesterday, basically telling Sarah what a contribution she's made. But he has left a legacy for Maury Gertz that you saw, who left a legacy for me, who I came after. And now you're going to meet Jitma, who's the next generation coming after. And it's not just those 4. There are many others like that. The same is true at Dana-Farber, the same is true at MD Anderson, the same is true in Australia, the same is true in the UK. But it's all about the next generation bringing those people along. So how do you do that? Well, the idea is people that are excited about Waldenstroms come up with a proposal of what they would do. They're engaged in the field. Those get selected. They get funded for over 2 years to focus on Waldenstroms. And currently there are 4 Kyle Career Award projects that are being performed. So I'm going to introduce the first of those recipients from 2023. There are more that are going to be adjudicated this week. I'm going to introduce you to Dr. Abeykoon. He's going to come up in just a second. He's in the Division of Hematology at Mayo Clinic. He's an assistant professor there. He originally trained at the University of Kansas, then did medicine and oncology, hematology oncology at Mayo Clinic. He focuses on DNA repair mechanisms, and he's going to tell you a little bit more about that. But the goal is obviously to work out how cancer cells can fix their DNA and how we can maybe potentially disrupt that. So I'm going to have him come up, tell you a little bit about himself and a little bit more about what he's doing. And then I'll come back up and introduce the next person right after.
Hello, good morning, everyone, and thank you for the introduction. First of all, I would like to thank the IWMF and the organizations in supporting us, supporting the next generation, and also funding our research so that we can contribute, we can deliver meaningful contributions to our patients. So my research got funded last year. The title is Defining the Prognostic Significance of TP53 Alteration in Waldenstroms, and use this for therapeutic benefit. So TP53 is the guardian of the genome. So TP53 is the policeman. So meaning that if the genome misbehaves, it fixes and let the cells survive. Normal cells need TP53. And we know that TP53 mutated or altered cancers behave very badly. That means they are more aggressive, does not respond to treatment, and so forth. So these cancers are in solid tumors, the breast cancer, the pancreatic cancer that you commonly hear, and also in blood cancers too, such as leukemias, CLL, and large cell, the lymphomas, and so forth. However, the impact of TP53 in Waldenstrom's is unknown. And also, we don't know the prevalence. Previous research has discovered some, but the prevalence is all over the place. Some says 3%, some says 30%. We don't know exactly the prevalence. And also, we don't know the impact of this alteration or mutation in our patients and how this behaves with how this disease responds to treatment. So that is the reason for this grant, and also not only to look at the impact of TP53, but in Waldenstrom's, but also to see how we can use TP53 as a marker and as a biomarker, and also how we can treat TP53 mutated cancers better down the road. So we had two aims. First aim is to use a large cohort of patients that, thanks to Dr. Ansell and his team, we have a big biobank at Mayo Clinic, so that we are equipped to answer these questions. So to use a large cohort of patients and do sequencing, like what Dr. Ansell mentioned, the omics part, the signaling part of that roadmap, to answer the question, how many of these patients have TP53, the mutation altered, gene altered in their disease. And also, not only that, we will be able to answer the question, how does these patients survive and how do they live? What is the response to the disease, to the treatment when they have the TP53 alteration? So that is the first question, first aim. The second aim is to see if the patient has a TP53 mutated Waldenstrom's, can we use a different treatment approach to achieve a better outcome for our patients? So that is the aim too. So the first hypothesis is that TP53 mutated Waldenstrom will be aggressive and lead to a poor prognosis. And the second hypothesis is that when TP53 is mutated in Waldenstrom, these cells are very, 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 very aggressive. And they actually use the DNA damage repair pathways more than the TP53 unmutated cells to sustain their survival. So then we ask the question, can we target these DNA damage repair pathways to impact the survival of these cancer cells, TP53 altered cells, so that our patients achieve a better good benefit or a better outcome if they have TP53 altered cancer. So this is the timeline I just want to put forward, that we are doing as we expected. So we have just submitted the samples for sequencing. We will have data soon, and also aim 2 that we are working on track for completion. So we will be able to get the data and the outcomes and the results for you with our research at the end of the mid of 2025. So again, thank you for funding us, and we will update with the results in coming meetings. Thank you.
So we're actually very fortunate to have the second of the 2023 recipients here, and this is Dr. Guerrara. She's an instructor in medicine at Harvard and is a physician scientist looking at diagnosis and treatment of various hematological cancer. She's going to tell us a little bit about her heritage from Italy, and has been now part of the Bing Center for Waldenstrom's, and focuses substantially on uncovering mechanisms for why cancer cells in Waldenstrom's are cancerous, how they evolve, and maybe how we can target them. So she's going to tell us more.
Good morning, everyone. Thank you so much for this opportunity, for giving me the possibility to actually show you my research and for funding me. I'm very, very excited. And so I asked myself how I wanted to tell you my story. So I thought to take you on a journey through history and around the world, which is a journey of exploration. So let's get it started. So please, ladies and gentlemen, fasten your seatbelt. We are ready to start on this journey from my birthplace, which is Sicily in Italy. Can we put this back, please? Okay. So I've chosen to start with this clip to give you a glimpse about the depth of diversity and history from the Sicilian Greek, Latin, Arabic, French, and Hispanic periods that I've been surrounded with since I was a kid. And this is important for me because Sicily was the first place that taught me the power of complexity and an essential skill for a scientist, which is the ability to step back and observe. So as you can see, as one of 4 girls, and being shy, I actually had to learn pretty fast the skill of survival. But also my family taught me a lot of other important concepts in my life: love, fun, the importance of family and community, belonging, and the importance of also of exploring and doing different experiences. And also the place I come from sits in the middle of the Mediterranean Sea and overlooks on the other side Calabria, so the rest of Italy. So that taught me always to look beyond and use my curiosity and imagination. So as I grew older, I decided in my high school to take on humanistic studies. And this is where I encountered ancient Greek and Latin and philosophy and art history, Italian literature and theater. And all of these languages taught me my roots, and also that there are different languages that can be spoken that are within me, including the language of my body. So there is a quote that I always held really close to my heart, which are the words that Ulysses says in Dante Alighieri Divina Commedia, where he says, "Considerate la vostra semenza, fatti non foste a viver come bruti, ma per seguire virtù e conoscenza." Meaning, consider the seeds from which you sprang. You were not made to live like brutes, but for pursuit of virtue and knowledge. And that has been always like a light for me. So as I then grew much older, I decided to move on to pursue my dream career since I was a kid, which was becoming a doctor. And I did it moving to the north of Italy, to Pavia. So this was my first migration of a long series, now it's been 20 years. So this university is very known, and because counts among many others between their teachers and their scholars, some great presences like Alessandro Volta, the inventor of the electric battery, Camillo Golgi, a Nobel Prize for Medicine for his studies on the central nervous system, Adolfo Ferrata and Eduardo Storti, who were pioneers in the research for acute myeloid leukemias. And so those were great inspiration that I took throughout my training between Italy and Germany. But I also always hold very close to my heart the teachings of another Nobel Prize for Medicine, Rita Levi-Montalcini, who was a brilliant woman who achieved incredible results in a terrible time during World War, and in a moment, in an historical moment, where women were precluded any kind of high-ranking careers. And here we fast forward to my third year of med school, where I fell in love with hematology, which was when I actually learned the potential, the great potential of science, and how the discovery of Gleevec, imatinib, completely changes the disease, the story for patients affected by chronic myelogenous leukemia, by targeting the tumor-promoting effects driven by the protein fusion that is pathologically generated in this disease, the BCR-ABL complex. And there we go. I decided to espouse hematology, and then I went on with my clinical fellowship, always in Pavia, where I had 5 years of clinical training, and I encountered a variety of different range of hematological diseases and patients, and I had great incredible human experiences with all sorts of patients, and also great mentors. Among them, Marzia Verettoni, that you see here in the picture, and Luca Arcaini, who actually created the bridge for me to come back here to Boston, and actually started joining the Being Center for the first time in 2016 with my postdoctoral research fellowship, and now with my instructorship in medicine. And I've been super lucky and fortunate to be in an environment, we always talk about right environment, so it was really conducive environment to learn and to just great inspiration from my great mentors. And from them, I've started with my first scientific work, focusing mainly on the pathways of MYD88-mutated disease, and focusing on two big parts, the genomic part, going into the depth of the 6q deletions in the MYD88-mutated disease, as well as the impact of MYD88 by itself in a mouse model. And so if I were to summarize what I've learned so far in my life, I would say I learned the power of complexity, which gave me the possibility to observe, and then through observation, changing perspective, and then coming back to the same complexity and having a different point of view. And now I would like to take you with me on this inward journey into complexity. So from the outer world, let's get into what we are, because we are deeply and fundamentally very complex in a beautiful fashion. So yesterday, possibly a lot of you heard from Zach Cantor, his great lecture, so I'm not going to even try to reach his level, but just to summarize a few concepts. Our genetic material is stored in our cells in the form of DNA. Then RNA, so the genes, actually have all the information that serve them to be translated into proteins. They are really the true actors of any biological mechanisms into our body and function. But I would like to tell you a little bit further these stories about the RNA. So the RNA is not ready. It needs to go through some, changes before it becomes ready to be translated into protein. So this process is called splicing. So as you can see here, there are these main pieces of the RNA, which are all these boxes in different colors. And in this process of the splicing, these different pieces gets assembled in different way, from the pre-RNA into what is now called alternative spliced mRNA, and I like to call it RNA flavors, okay? So there are different flavors in which this RNA comes with, and this gets translated into different proteins. So from the same gene, we can have proteins that have different, very different functions. And this is relevant to my Robert Kyle Award now, that focuses on this gene, which is called Wing2. We got really excited about this gene a few years back in our research because we realized it was one of the top dysregulated genes within the mediated mutated WM. And we also noticed, thanks to the recent ZAC work has pioneered, that it is really upregulated early in the disease. So you can see here where you see HD, those are healthy donors. So healthy donors have zero expression of the gene. So this gene is not supposed to be there. But if you look at pre-BCL and pre-PCL, which are early stages of the disease, you see that the expression goes aberrantly up. So why is that? So our question is this a gene that is important to be turned on early on to favor the malignant transformation of mediated mutated cells? So these are the RNA flavors that Wing2 comes in. So those are the ones that are actually known. But thanks to the work that you guys funded, we found many different other RNA flavors of Wing2. And especially we noticed that in all the samples, these two pieces that I highlighted here are missing in Wing2 in WM patients. So not only this gene is expressed, but these pieces are missing. So why is this the case? So we focused on that one form, the one new RNA flavor that we discovered, to try to do a model, a cell model, to try to see in a cell system what happens when we have a lot of these pieces. And we also created another form where we reintroduced back the two pieces missing to see whether this could help the cells or not. And in fact, what we discovered is that the addition of that novel piece that we call Wing2 PB4 is actually beneficial to the cells to grow. So the cells that have high levels, the tumor cells that have high levels of this protein, are actually those that have an advantage in growth and exert many different signals that are known for B cells to be, for Waldenstrom cells to be important. So thank you to the work that you are still funding. We are continuing to understand how this gene works and how we can therapeutically target it. And this is particularly important because in other diseases, this gene has been described having the opposite effect, yet that of suppressing the cell, the tumor, while in our case it seems to promote. So with this one, I would like to thank you all really for this great opportunity, for trusting me and trusting my science. I would like to really thank all of you, all of your family, because I know how important that is in the setting. I would love to remember here Glenn Cantor, which I had the great opportunity to meet, and then thank my mentors, Steve Trion and Zachary Hunter, and once again, thank this long-standing friend of complexity. And I wish for all of us that we can actually embrace complexity within ourselves and also in the outer world, and embrace it with curiosity rather than fear in an historical moment where they tell us that complexity is an enemy rather than a great resource. Thank you.
Well, thank you very much. I thought Renton was kind of nice, but I'm liking Sicily quite a lot there. So just want to take us back for a second. Because, every so often it's important to link things together. So remember how I said we didn't have any tools at all? So you might see there BCWM.1, MWCL11. Those are two cell lines made out of that initial meeting where we said we didn't have tools. So now we have tools. And all of this work that you see understanding the proteins and which ones go up and which ones go down are thanks to you, because those cell lines didn't exist 20 years ago, and those are cell lines that really show us and help us understand Waldenstrom's. All right, so I just wanted to end now with what else could we do? So again, just to complement the International Waldenstrom's Macroglobulinemia Foundation and all of the ways in which you're supporting the next generation. And you just heard from two of the budding stars, if you like, and the future of Waldenstrom's research. But one of the additional ways there are real opportunities for us to do that is to continue to support young investigators. And just you've heard mention of Glenn. Many of you know Glenn well. He was part of the Board of Trustees when joining in 2020. served on the SAC committee, that's the Scientific Advisory Committee, and also on the Grant Review Committee, and was involved in the Torch. But his passion was really about how to support the next generation. So we're really excited, and we want to really thank the family for their support in this area, and that is to announce a Memorial Early Career Researcher Fund in honor of Glenn Cantor. And this is and this is all about providing additional ways to engage and bring along the next generation. So you heard a lot about scientists and the next generation, but clinicians and the next generation are also important. The people that will treat the next population of patients who are unfortunately getting this disease, but which hopefully in the not very distant future will be cured of this disease. So I just wanted to say thank you so much to the family. Here's a photograph of Glenn and his family. And again, just remember him. But this fund will be an opportunity to provide fellowships where people can come and train and learn about the clinical ways in which one can manage Waldenstrom's macroglobulinemia. Thank you very much for giving us a chance to just review with you some of the research initiatives. And thank you most of all for your support. Very grateful.